So these are the, you have to count the cells. Today it is about counting WBC. This is the counting grid. If you have read counting grid has this thing. This is the whole square. In the counting grid Neubauer's chamber, it has two grid, and don't call it the Neubauer's chamber, call it strictly the improved Neubauer's chamber, because in case of the, if you have read it, so as you did not attend, I had asked many of you to inform others. There is no CR till now, to inform others to read it and come. This is the Neubauer's chamber, the upper part and the lower part, beside above the horizontal group, two identical counting chambers. We can count in any of them. Ideally, again, counting in both and making the average and all, though we are talking of uniform distribution during charging the chamber. Over this chamber, a cover slip will be put, and cover slip, in between the cover slip and the counting grid, the blood will be charged. So that is very important, to charge the blood correctly, which you have to just learn by experience, that means do hands on and learn how to charge the blood correctly, so that it is uniformly distributed. So the blood will be distributed in the whole of the counting grid. In this RBC, this is the central square of the counting grid where RBCs are counted, and see, these are squares. 4 squares are there which are not for counting cells. So they have been dissected and bisected and dissected by triple lines, which are dividing the RBC square. The large RBC square is divided into 5 into 5, 25 medium RBC squares, and the triple lines are dividing each of the medium side RBC square into smaller. Each of the 25 is divided into 16 each smaller RBC squares. So the RBC squares are of, we should not say 3 types, because the same square is divided into smaller divisions, not 3 types, but we may say 3 types of divisions. So, or the naming of the squares according to these divisions: large RBC square, medium RBC square, small RBC square. But for the counting the WBCs, we use the 4 corner squares of the grid. They are just divided into large and small, nothing medium there. Large RBC square, and the 4 into 4, 16 in each is the smaller RBC square. So during the counting the cells, you will require the measurements. So each counting grid as a whole is 3 into 3 millimeter, that is, the area is 9 square millimeter. For each WBC square, it is 1 into 1 square millimeter. Now WBCs are less in number, and dilution of blood is also required to a lesser extent. It is diluted just 20 times. How it is, we can explain, and we have to explain how, but it is diluted just 20 times. And I think, seeing it under the low power of the microscope, that is the 10x lens, okay, low power of the microscope, you know what is the low. There are 3 types of lenses for you. You are the student who joined on 6th January. So in the microscope, we'll just see there are 3 lenses, 3 objective lenses. One is magnifying the object 10 times, one is magnifying 45 times, the other one is 100 times. For WBC counting, usually the low power is sufficient, and magnifying it 10 times, you can count the cells. WBCs are lesser in number, much lesser in number than RBCs. Only 1:20 dilution is required. I mean 20 times dilution of blood is required. Without diluting, what is the purpose of dilution? Because so many cells to be counted, it won't be possible. So without diluting fluids are there. So for WBC, today's class WBC, it is the Turk's fluid, which contains glacial acetic acid and Gentian Violet, and distilled water, obviously distilled water to make up the solution. Glacial acetic acid lyses the membranes of RBCs and WB, all cellular components. In some books it is written glacial acetic acid lyses the RBCs, but I think that is not correct. It lyses the membranes, but as WBCs have a nucleus, and it can't be, he has not seen, but rest of you have seen, it is not like Leishman staining where it will be seen as lobes of nucleus. Here one rounded nucleus, if you see, you count it as one cell. So this will be possible under the low power of the microscope, and you can count like this, that this is, here is say one cell, here is one cell, so number of cells present. And for the calculation, you will require these measurements: 1 into 1 square millimeter each WBC square. And what is the depth? So you require a volume. So what is the idea? The volume of the squares is the volume of the blood. Now in a 2-dimensional surface, it is not, the word volume can't be used. So volume is for a three-dimensional. So there can be a volume of this room, but there cannot be a volume of the floor or the roof. Okay, strictly speaking, because it has no depth or height, that component, that physical parameter is not there. So what is actually the volume, how it comes? The covered slip is placed over the counting grid, blood is charged there. So the volume of the blood includes the just small space between this covered slip and the counting grid. This can be called that height, what we are calling height. Length into breadth into height is the volume. This height is the space between the covered slip and the counting grid, that is 1 by 10 millimeter, and the counting grid, this one is 1 millimeter, this is the 1 millimeter. So the volume of blood overlying these squares. So very roughly it is written in books volume of the squares, but actually strictly speaking it is not the volume of the square, it is the volume of the blood overlying the square after charging the blood. Considering uniform distribution, so the blood overlying the square, volume of the blood will be 1 millimeter into 1 millimeter into 1 by 10 millimeter. So if in that volume certain number of cells are present, we can calculate in 1 millimeter cube how many cells are present. This is the principle. So what is the volume of the not 4 squares strictly, it is written volume of 4 such squares, but volume of the blood overlying those squares, it is 1 into 1 into 1 by 10 is the volume of 1 WBC square. So for 4 WBC square, it is 4 into 1 into 1 into 1 by 10, that is 2 by 5, and don't take it as 0.4. I will suggest you not to. When you make calculations, considering it as decimals, you may face problems. So it is easier to calculate it as 2 by 5. So 2 by 5 cubic millimeter contains certain number which we will count. Say we just say this as X, capital X or capital N. So 2 by 5 contains certain X number of cells. So 1 millimeter cube contains 5x by 2, x by 2 by 5, 5x by 2 number of cells. So 1 millimeter cube, this is after dilution of the blood. Dilution of the blood is done before charging. So dilution of the blood, so in case of WBC it is diluted 20 times. This is the WBC pipette. Blood is taken up to this 0.5. Again this has to be explained. 0.5 have no units of volume. So this is what was done in the first session on this topic. 0.5, it is not milliliter or microliter. In hemoglobin pipette you took blood up to 20 microliter. If up to some pipettes there are 10 microliter, so you have to take it twice. But 20 microliter, it is not about microliter here, it is just parts of volume. Any part, this 0.5 can be say 4 microliter, 5 microliter. So, but if 0.5 is say, I am just assuming one figure, 4 microliter, then this 1 means 8 microliter, double of that. Here in above the this bulb, this is called the bulb of the pipette. Above the bulb there is a marking called 11. So 11 marking means it is 22 times of 0.5, the volume. So if 0.5 is say this, this is arbitrary figure, 4 milliliter, then 22 times of that is up to 11, that is 4 into 22. That is just an arbitrary figure. So these are not 0.5 microliter, remember that. This is just parts of volume. We don't consider any particular unit of volume while taking the blood. We take blood up to 0.5, this is just for how many times the blood is diluted. 0.5 blood, then suck the fluid will push the blood up in the bulb, and the bulb and the fluid just allow to rub you with your hand very gently for say maximum 1 minute, so that the blood and the fluid is mixed in the bulb. So below the bulb, this is called the stem of the pipette. This is called the whole actually strictly speaking the stem should be called above the bulb also, but strictly speaking between the stem towards this upper end is the bulb, but strictly casually we say this is the stem of the bulb, the lower part of the stem. This has no blood. After when you go to charge, blood and fluid has got mixed within the bulb, not below the bulb. So below the bulb, that fluid has to be by blowing out, that may be simply maximum 1 to 2 drops. This very gently and carefully this part is blown out, because that does not contain any cells. Blood and fluid has got mixed in the bulb. When we have drawn the blood and then drawn the fluid has pushed up the blood in the bulb. So this has to be discarded very carefully, so that blood does not itself, the diluted blood does not go out. So this is discarded and then the rest is charged within the Neubauer chamber. So that blood is why we are calling it 20 times diluted, because the whole blood is this blood and fluid mixture is 11 minus 1. This 1 does not contain any blood cells. So 11 minus 1 is the total solution where we will get, or the diluted blood. But out of that, how much is blood? It's only 0.5 parts of volume of blood. So 10 parts of volume of this diluted blood, but only 0.5 is actually undiluted blood. So the dilution has occurred 10 by 0.5 times. If by mistake or even intentionally we take blood up to 1, then blood will be diluted only 10 times. Now lesser the dilution, mathematically results can be expressed, but it will be difficult in counting the cells and all. So this has been standardized. For RBC pipette, the blood RBC counting, the blood is diluted 200 times. Now theoretically you can dilute 100 times or 50 times or whatever, but as the cells are so huge in number, diluting will lesser dilution, cell counting won't be possible practically. Okay, so we dilute it as per the standardized method. We take blood up to 0.5, but if blood is taken up to 1, mathematically it's diluted 10 times and the result should come, provided we can count the cells. If cells, if it is not diluted, cells will become, under the grid it will become very congested if only blood is there. So that is the purpose of dilution. So it is diluted 20 times for WBCs. So when we write 1 millimeter cube of the diluted blood contains 5x by 2 number of WBCs. So 1 millimeter cube of undiluted, or the blood which was actually taken, blood sample which was taken for the our experiment purpose. My purpose is to find out the number of cells in 1 millimeter cube in that blood. That contains 5x by 2 into that dilution factor 20. So it comes to 5x by 2, 50x. So put x at the last and multiply it by 50x. That is your count. That is the count of WBCs per millimeter cube of blood, or same thing is microliter of blood. 1 millimeter cube is 1 microliter, as we know theoretically. So this is the principle of the experiment, how we do it, and principle and the procedure. RBCs calculation are just a bit more complex, slightly more complex than these, with large, medium and small squares. Those who don't know still now, we may discuss it when we do RBCs again the next week. So today you do the WBCs and this will take time. These WBC squares can be named as left upper square, right upper as per the position, left upper, right lower, left lower, like this. And also the RBC squares can be named so. Each square when will be needed, you have to focus yourself. Like if you have to focus the, say, left upper WBC square, just think. Now after you have seen and read once or twice, you have known about the topic, just think. The left upper WBC square, how to identify? 4 by 4 box, it's okay, but how will you identify that it is the left upper or the right upper, or the left lower or the right lower? 4 by 4 box is just this WBC square. So on the simple things, just look at this, how to identify looking under the microscope. Don't say theoretical. Like in DLC, you can't say, in DLC means during identification of blood cells, you can't say, okay sir, this is a lymphocyte because this is 12 micrometer in diameter. You are not measuring with a slide calipers over there under the microscope. You have to compare with the size of the WBCs, RBCs. Here, how will you tell whether that square is the left upper one? So just see, so these should come to the mind, common sense. So when you will look, you will see, say the left upper, to the left and the top, there is nothing. Nothing means nothing. So there is no counting grid, left and the top, but to the right and the below there will be triple lines. So that confirms it is the left upper square. All 4 WBC squares will look similar, but how you do you focus? You say that I have focused the left upper one. So this is somewhat a bit more, I should say, not complex, but in case of this RBC squares counting, these has rules. Not all RBC squares has to be counted cells. Cells are counted in this corner 4 and the central one of the central square after dividing into large, medium, small. RBCs are counted in the corner, medium squares and the central one. So how to identify the corner, this left upper RBC square, left upper this medium size RBC square. So there will be triple lines to the left and the top and similar RBC squares on the other sides. So that confirms that that is the left upper. Now from the left upper, if you move 3 squares, you get the right upper and confirm it by to the right and the top there are triple lines, and to the left, to the below and the left there are similar looking RBC squares. During the central RBC square, there will be similar looking RBC squares on all sides. So how do you confirm it's central? It is actually by going on counting. You move from here to here, here to here, move 3 cells. After all the corners are completed, conventionally it is done in the clockwise, this is clockwise fashion, and at last from the right lower one you go 2 up and 2 into the middle. That is for the RBCs. So today concentrate on WBC. So just you have to, because if you have, what is the purpose of identifying the left upper square? You will count all 4. In our case of RBCs, you will count the medium squares only 5, not all, or not all will not won't be possible. But here you will count all 4. So what is the purpose? What is the aim? There should be a purpose of identifying. Why should I identify? As I will count all 4, is there any need to identify? Suppose you have randomly focused any one of them. So what is the need to identify? You will need to know that in which direction I have to move next. If you don't know, even if you have reached at one WBC square randomly, reaching it randomly is first not encouraged. You have to reach by knowing from the RBC square. When you focus the microscope, then you so I am moving in which direction. So move in one direction and focus one, then you know in your mind that this is the left upper. So I will move now to the right upper. Conventionally it is done clockwise. You may do in any direction, but conventionally it is done clockwise. Left upper you move right upper. You will cross this square and then reach it. Cross this square and reach it. Then last, after the 4 are completed, in the case of WBC there is no more complication, the 4 squares are completed. Okay. So that 50x will be the last figure. So what should not be said on record is that you expect how many WBCs per millimeter cube of blood? 4,000 to 11,000. That is, this is the normally accepted value. So if say it is 8,000, okay, so if it is 8,000, say 8,000, so then x should be, if it is between 4,000 to 11,000, that x should be between 80 and, for 11,000 it has to be 220, right? So between 80 and 220, but this is just, this should not be, that is back calculation. So x is to be expected like that. If you are to, as we are taking our own blood uniform distribution, so x is expected like that, but maybe that whatever comes you have to write. So if x has to be expected, you are counting 4 squares. Say if x has to be 100, then roughly it will be 25. Don't show 25 for all squares. So it can't be that actually that uniform. So it may be 30 in some, 40 in some, 20 in some, like this. So that is expected. That is calculating from the backwards what is expected. But whatever you show, but if x is say instead of 220 it comes 600, that is abnormal count. That means you are counting wrongly. Or if it is just few, where you say x comes to about say only 50, that is not correct, or even below that 40, 30, only 4 or 5 cells in one WBC square. That means one large one. So that is actually, the process is wrong, because you are doing with your own blood. Expected we are all healthy persons, so that the count should be in the normal range. So remember that at the back of your mind, but don't try to do like that, the examiner will catch you. Especially if you write 20, say 25, 30 in all squares, 25 for this square, same number in all squares. So it has to be done, it has to be shown, it has to be focused on the mind, it has to remain focused on the microscope. The examiner has the examiner may, what he or she may do, Mr. just focus it himself or herself and show that how many cells, how you have counted the cells. Focus, show me the cells, like this he or she may say. So you have to do it properly and then arrive at the result. So in the laboratory notebook, you will write principle, procedure, calculation, result. Okay. Aim of the experiment, principle, procedure, calculation and result. Like this, and whatever result you get here, write. If you copy from others, it will be the same result in all copies. Don't copy from others. You, whatever you get, you record that in your notebook and write that in your practical. How do you differentiate between the RBC pipette and the WBC pipette? Only the color. The color may fade away. Even this, not easy to fade away. These fades away. These are relatively, I think, new instruments. In older instruments, see the markings, see the size of the bulb. That is the main size of the bulb. Compare the size of the bulb. And markings, you see 101, 11. So you have got seated according. Seated means you are working according to the roll numbers.
Yes. So I just want to find out. So here, exactly where should I put the mixed blend?
So you will do it. After you put the cover slip, it has to be put under the cover slip and the Neubauer chamber.
Okay.
Such that it is actually, it will be uniformly distributed on both the upper grid and the lower grid.
Okay.
But during counting, we use any one of the grids. Considering distribution is uniform. It has to be not to be done separately. When you give, it will automatically it will go to both the grids, spread to both the grids.
And cover slip, we have to apply.
Cover slip, the ideal is to apply the cover slip first, because what will happen if you apply blood first and then put the cover slip, the blood will be spill. So very carefully it has to be given between the cover slip and the, after putting the cover slip, in that small gap the blood has to be, you have to put with the pipette. The lab technician will help you. Achha, who is the student who joined just recently? These are lenses. This is the eyepiece lens. Okay, so the magnification of the object will be by this lens and this lens together. So these are called the objective lenses. There are three types. One magnifies it 10x. Let's bring this to that table. So even this is to be learned, how to hold the microscope. Have you held the microscope before? So it has to be held by supporting with your non-dominant hand. Usually right hand is your dominant hand. Yours is right hand. So you support with the left hand about the microscope. These microscope, we have to take them beside the window and all, so that the daylight source. So this is the light source here. So I'll just tell you, this is simple, that focusing, but not that simple. So this is the quartz adjustment screws for bringing the platform, this platform up and down. This is the fine adjustment screw. After something is focused, it will give you just fine tuning, just to make it a bit clearer. These are the eyepiece lenses which can be detached. Now for these types of microscopes, you have to practice with two eyes. If you have seen in the microscope that there are, in those microscopes where there is one eyepiece, you have to just learn closing one eye and see with the other eye. But here, after a few days, our eyes have binocular vision. You will see the same thing, but you have to, it will just take a bit of time to adjust seeing with the two eyes together. This magnifies, if it is, say, these are the different objective lenses. Say if the 10x lens is used, the 10x means magnification 10 times, and this is magnifying the object 10 times. So total magnification is 10 into 10, 100 times. And how to use the light source and all, how to use this is called something called the condenser. See, the condenser is going up and down with the help of this screw. This is another screw for adjusting the position of the condenser. This is for the adjusting the focus. This and this for fine tuning the focus. This is the coarse adjustment screw. This is the fine adjustment screw. But this is for adjusting the condenser. When the 10x lens is used, condenser should be lowest in position for better focus and better this lighting, the light source, the light to be entered and focus. When the condenser is, this is, yes, 10x. When the condenser is at 45x, it should be somewhere in the middle, just you have to just do the, you have to practice, and by that experience it comes where the light source is okay and you can focus correctly. For 100x the condenser is in the highest position. For 100x we use something else called the, for better magnification 100 times we use oil, that is we use during differential leukocyte count, which you have not performed. We'll see what we can do about that. But microscope you will need in this experiment also, and the first class was on microscope. Just ask your friends or colleagues that what are the topics to be written. You will get some time to write those. You see the notebooks of your colleagues, they are already writing those. So they are the topics in order. And let's see, we'll talk to the HOD about what can be done for those who have joined recently, how to cover those topics. But theory topics can be covered. That means theory topics can be repeated. These things we will try to arrange some classes for those who have joined so late. We'll try. Okay, you, and for the rest of the departments, anatomy and biochemistry, you talk to the HOD. Here I'll talk to the HOD say next week about this, that you have joined one, somebody told yesterday joined on 3rd January. So do you know how many are there like this in this out of 150, how many joined? Yes, after this last round. How many? I think maybe less than 10.
Actually, I don't know about the management quota I don't know, but there is one from state quota and I got the ticket.
Okay.
Only one from state
Less than 10. Where are you from?
quota.
Kolkata where?
Guria.
Goria. Okay, you carry on. We'll help. This is the, these are the, probably 7, 6 or 7. Okay, this is the diluting fluid. This is the RBC pipette and the WBC pipette. These are pipettes. Pipettes are this, we suck blood or fluids with the help of pipettes. These will be identified according to the size of the bulb. See here also the color of the bead has faded away. So don't look for the red bead. In the RBC pipette usually the bead inside the bulb, this small thing bead is usually red in color, but it fades away with time. So you have to identify with the size of the bulb, and the marking is 101. Markings may also fade away, but marking is 101. So the size of the bulb, see, actually 101 means it is this portion containing 100 times volume of this portion. So, but this is only 10 times. Bulb will always be larger in the RBC pipette. In the same hemocytometer box, you can compare and see. So for RBC pipette, RBC counting, dilution required is much more. So today you have to work with this WBC pipette. Okay. And the diluting fluid is the Turk's fluid, which contains glacial acetic acid and Gentian Violet. দেখো 0.1, 0.2, 0.3, 0.4, 0.5 this one, তারপর 0.6, 0.7, 0.8, 0.9, 1.
আচ্ছা যখন আমরা ফেলবো তখন এই 1 পর্যন্ত রেখে মানে থাকবে পাইপে যখন আমরা
এই হ্যাঁ এইটুকু ফেলে দিতে
ঠিক
হবে।
আছে থ্যাংক ইউ
Study the topic and come, open the book. So now you have done it, you should do it immediately, no? So follow the procedure at least as beginners. So how to do? See, wipe of the blood, wipe of the first drop of blood, hold the pipette horizontally, apply it steep to the, taking care that there should be no air bubbles. That is, immediately thereafter holding the pipette in a nearly horizontal position, then fluid is sucked up to the pipette and exactly up to 101 mark, that's okay. Pipette is then, it is gently rotated for 1 minute, this give a suspension. Now to so these steps, you have to do these steps. So after charging the blood, I'll just suggest don't put the Neubauer chamber over, focus it first and then use the blood. Okay, first focus it. So between the, when you will keep it over here and then, because after you take the blood in the Neubauer chamber, then if you go to focus it, this blood may again spill out. Okay, after focusing, this have to be gently done from the sides between this. See written, focus the, discuss the first few drops of fluid from the RBC pipette. They constitute the unmixed help fluid. Charging the Neubauer chamber, small drop of blood mixed with fluid is allowed to form at the tip of the pipette, and this drop is gently brought in contact with the edge of the proper slip at an angle of 45 degree with the horizontal. Okay, here the fluid is drawn into the chamber by the fluid is drawn into the chamber by capillary action. So still it is still better if you can keep the chamber over here focused or not in focus, and then don't move the chamber much after, or you have to do it very carefully. After putting the blood you have to put it then very carefully and then focus. Okay, হ্যাঁ আরেকবার পিক করতে হবে। ল্যান্সেটটা কোথায়, যার আঙুলে প্রিক করেছে, হ্যাঁ তাহলে সেই করো, না হলে ভেনাস ব্লাড নিয়ে করতে হবে কিন্তু মানে এক্সপেরিমেন্টটা করার জন্য।